name's Boston Page, and for the Science and Engineering Fair, I made a cooling shirt. The problem I identified was that California gets very hot and it makes many people very uncomfortable, so I made this shirt to combat the heat. It's made out of a silk blouse, mesh, and fans. The use of the silk is that it makes you feel cooler. It's been proven. And the use of the fans is to create an airflow similar to the wind that actually causes you to feel like the temperature is actually cooler than it is. And mesh pockets, airflow through the mesh similar to that of an air hockey table. How to make it's very basic. All you have to do is get silk blouse, sew in pockets on either side of the blouse, and then insert the fans into said pockets. I met my constraints being that it had to be relatively comfortable, able to charge instead of requiring batteries, and that it had to make the wearer feel significantly cooler, which it did, and the constraints being that it must cost less than $70 to make, and that it must be made only of mesh, silk shirt, 2 fans, and the necessary stitching materials to combine them. I would consider this a success, and thank you for listening to my presentation.
Hi, I'm Jordan Kruger, and my science fair project was where I tested the genotypes of these 3 succulent plants and compared them to what the genotype was of what the offspring would be using a method called Punnett squares. Punnett squares is where you predict the traits of the offspring using the traits given by the parent plant or anything with the traits that you're comparing. I did this to answer the question of whether the genotype of a cutting or clone of the parent plant is the same as the genotype of the original plant. In this case, the parent plants are the 3 different succulents who all have their own Punnett squared, and the 3 different succulents all came from the same original parent plant as well. So what I got the idea from was I've had this idea in the back of my mind. I don't know if I saw it at the last year's STEM fair, or we might have done it in my science class in 8th grade, where we learned about DNA in strawberries, removing the DNA of strawberries, comparing the DNA of strawberries to other stuff, and manipulating it, testing it, pulling it out. And then also, at the time when I found this out, with that in mind and interest, we learned about genomes and biology. And I wanted to be able to combine the 2 using my love of plants. I'm taking the horticulture pathway at my school, so I wanted to be able to combine all 3 of these, and that's how I came up with this project idea. What's interesting the most to me, why this appeals to my interest, is, like I said, my horticulture pathway and me loving plants, and also the fact that it's an investigation, figuring out the genotype, figuring out if they're the same, if they're different, and I'm also interested in investigation, stuff like that. So being able to combine those 2 interests, this made this project so fun and educational for me that it was just amazing. I think this is important for the real world because nurseries use a method called cloning, where they take cuttings off of plants, the original parent plant as they call it, and replant them and regrow them and use those to sell as a reproduction method so they can get more of the plant. This reproduction method is not bad, and there's nothing wrong with this. It's just to show that this experiment is to show the consumer, because if I wanted to be a consumer myself, is to show the consumer if they want a specific plant and they look at the parent plant and are given a cutting, that it might not turn out the exact way that they saw. However, the traits of the plants do not affect the condition that the plant needs to be growing in. So, for example, my succulent, I water it every 10 days, and it's growing well. I keep it in adequate sunlight. It is an indoor plant for me. And so if I were to grow the cuttings, I would not change the living conditions at all just because the traits are different. My general result or conclusion was that the genotype of the offspring compared to the parent plant is inconsistent and the traits aren't always the same. On my Punnett square, one of my one of my traits did end up being all the same. These are all the same traits as the original parent plant, but the other two are not. So this shows that not all of the plants that are being cut or cloning are the same with the genotype. In this case, the clone is actually a leaf off of the succulent that can be dried out, placed in sunlight, and then just placed on top of the soil to be regrown. So yeah, that's pretty cool. I think what's distinctive about this project is being able to mix the different grade levels, and that it can be understood from a bunch of different, it can be interesting to older people, younger people, and being able to mix the two subjects. So I mixed science and I mixed horticulture, and also there's a broad range of stuff in there I was able to mix together to make this one project that can be interesting to multiple people, tons of people. To me, I find it interesting, but where I do think I could have done better in this experiment is I could have tested different types of plants. So instead of just doing the succulent, I could have done a bell pepper plant, taking and cutting off of that, or so on, just so because they're so different, to see if it's a different result. So if I wanted to compare the bell pepper plant, and if all of the clones were the same genotype, and that's just not the same as the succulent. So that's where I think I went wrong. I think I should have compared multiple plants instead of just the one. Even though I did 3 of them, it's still the same parent plant. So if I were to do the experiment again, I would definitely test multiple types of plants. And the question I have after doing this, that I think this led me to have, is does the condition or the environment of the plant affect the genotype of the offspring? So if I grew my succulent plant in different living conditions, maybe they were in a different pot, they weren't together, they had more sunlight, I watered them more often. If the environment affected the genotype of the offspring, and if they would be consistent. So thanks for watching my video. I hope you enjoyed my science fair project, and I had tons of fun doing it.
The idea for our project stems from a conversation my brother and I were having about the 4 main states of matter: solid, liquid, gas, and plasma. We realized that most people encounter the first 3 often in daily life. However, plasma is slightly more rare, and we decided we wanted to explore it further. The first thing that came to our minds was a Tesla coil.
The goal of our project, besides creating plasma, was to determine how the number of windings in the Tesla coil affected its output. Our hypothesis was that for the secondary coil, more windings will increase the strength of the electromagnetic field, and for the primary coil, we think that less windings will be better, but only up to a certain point.
We formulated our hypothesis based on the fact that a Tesla coil is a type of transformer. Because the voltage multiplication is equal to the number of the secondary coil's windings divided by the primary coil's windings, we thought that the less turns in the primary coil, the better. However, this only works up to a point. The reason being that the less coils there are in the primary, the less electromagnetic field induced. As a result, the secondary coil does not receive as much power. This means to get the best results, there should be as many secondary coils as possible. However, the primary coil will have an optimum number of turns, which may vary between applications.
Our project is important to today's society because every component in the Tesla coil is something critical to electronics today. Transformers are used to step up or down voltages and amperages, and are vital in moving energy around and making it usable to the devices in our houses. Most things do not actually need 120 volts of alternating current to operate. In fact, most electronics convert power to direct current before use. In solid-state Tesla coils, transistors are used. Without transistors, we would have almost none of our modern conveniences, certainly not computers as we know them, and civilization would be far less advanced.
Tesla coils create powerful electromagnetic fields, and we are very interested in the way the mechanism makes use of these fields to transfer energy wirelessly. Using S2 coils, it acts as an air core transformer. The electrons flowing through the primary coil create an electromagnetic field. Then, Due to Lenz's law and Faraday's principle of electromagnetic induction, the electromagnetic field excites electrons, which then flow through the secondary coil. The electrons in the secondary coil want to be grounded and have enough energy to arc down through the air. So much heat is lost in this process that the electrons ionize the air, turning it into a plasma.
One of the problems that we faced in developing our experiment was how to collect data. We didn't know a way to measure plasma, so we devised a method to measure the electromagnetic field generated by the Tesla coil. We created an additional coil with 200 windings. Applying the same concept that transfers power from the primary coil to the secondary coil, the third coil will transform the generated magnetic field into a current of its own that is detectable by a multimeter. This solution does create a new problem, however. The output of a Tesla coil is not a constant, steady flow of voltage, but instead fluctuates, and will not give a single measurement on our multimeter. This fluctuation can be smoothed out using capacitors and a diode, in our case, an LED. This made it much easier to get a result from our test coil. It essentially takes the average of the oscillations and gives a usable measurement. This works because the capacitors store excess voltage from power spikes, releasing it in the troughs, and the diodes ensure that the Tesla coil's output is direct current rather than alternating current.
A final obstacle we encountered during our first test was that our LED was being overloaded, causing it to fail. The real problem was that sometimes LEDs can suffer from partial failures that allow them to still light up. This was the case in our situation, so we thought our circuit was working, but it was not performing its job as a diode. Because of this, the capacitors smoothed out the sinusoidal wave in the alternating current, giving us readings that were close to zero. After reviewing the results and realizing that they did not make very much sense, we attempted to get readings again, but with another LED, and they were much more sensible. Before we restarted our tests, we decided to add an additional LED to our circuit in parallel, which would share the voltage equally so that we did not have to worry about them being destroyed and skewing our results.
To demonstrate our data, we created a line of best fit using the Desmos quadratic and linear regression feature. This essentially means that the computer, using the given function, finds the best possible fit from the data and displays it in the form of a graph, rather than just reading the data in its raw form.
From the graphs, we can conclude two things. The first four seem to show a parabola, which indicates that there's an optimum number of primary coils, in this case, around 9. The last three graphs show a linear relationship. Because these linear functions have a positive slope, the more the independent variable increases, the more the dependent variable increases. This means that the more secondary coils there are, the more efficient the Tesla coil will be.
We were able to support our hypothesis. We predicted that there would be an optimum number of primary coils, reasoning that less primary coils would step the voltage up further, but the electromagnetic field would have to be large enough to reach the secondary coil. The primary coil's graph line of best fit is a parabola, which means that there is an optimum number of primary coil windings, as we predicted. This data also seems to support our second hypothesis, which is that the more windings in the secondary coil, the more voltage our Tesla coil would output. The secondary coil graph shows a linear relationship, which indicates that the efficiency of our Tesla coil will increase proportionally to the number of windings in the secondary coil.
This was an incredibly fun learning experience, and it was fascinating to build a Tesla coil from scratch and see it.
Hi, my name is Coral Noonan, and I'm a 6th grade Red Track Santa Rosa Academy student. This is my science fair project, How Do Organic Seeds Affect Radish Root Length?
Where did you get the idea for your project?
I really like biology, and DNA and mutations in cells are really interesting to me. The actual idea, like the question for this project, changed quite a bit. It was originally how do organic seeds affect radish nutrient levels, but there are no nutrient tester kits available, so I had to do radish weight, and then I had to do radish overall weight because they weren't long enough, and then it switched to radish root length when I harvested them because I noticed a significant change in the length.
The length between the organic and the non-organic?
Yes.
Okay. What interests you the most about this subject that you chose?
I like biology a lot, and I think organic seeds are really important, and the non-organic seeds are getting affected by pesticides and herbicides and stuff like that. So I think bringing attention that there actually is a difference between the two, that's going to help.
So that was kind of the question I was going to ask you next was how will it help people today?
Well, it can help people realize that the herbicides and pesticides and genetically modifying plants is actually harming the plant itself. And my experiment shows that it actually does. And organic seeds, the root grew longer. So that's same amount of time, 42 days. That showed me that there is a difference. And the genetically modifying and using herbicides and pesticides, that's hurting the plant.
Okay.
Which in turn hurts the seeds.
Okay. And so that would be generally what your results and conclusions were.
Yes.
Okay. What do you think is special or distinctive about your project?
I don't think a lot of people are doing stuff with pesticides and herbicides and stuff like that. Also, I wasn't expecting to look at radish root length. I was going to do the weight, but when I harvested them, it was noticeable without even measuring. So,
And
that's different.
would you say that the goal of your project was, how would you describe
I
that?
wanted to see a difference between the two. So I wanted to see a difference between non-organic radishes and organic radishes.
And did you assume that the organic radish seeds would be healthier, so they would be better in some way?
I did. I thought that even if they would have been smaller or weighed less, I think that they would have, their nutrient quality would have been more because they're unharmed. They're basically untouched versus the non-organic seeds. They're being played with a little bit.
Okay. If you were to do this again, what would you do differently?
I think I would have planted my radish seeds earlier. So I think, because it was really cool to see the root length, but I want to see how the actual radish turns out. But I didn't get to do that, so.
Do you have any ideas why you didn't get the radish? Because they were 40 days, right? You had them for over 40 days?
42 days, and I'm not sure. I did not fertilize my soil. I took the soil that I had, and I have the full name of it on my digital science notebook, but I did not fertilize them. And so I think that was part of it. They probably missed about a week of sunlight because of cloudy or windy days, but I think that could have been part of it. But also, I think their growth time is like September to November and then March to May or something like that. So we are in California, but I think the weather was a little detrimental too, that it wasn't ideal for them. I brought them inside every night so they wouldn't frost, but I still think that the weather was a little bit of a change, or perhaps humidity or dryness in the air.
Okay. What problems did you encounter? You're kind of talking about that right now in developing or conducting your experiment. How did you overcome them?
They didn't mature in time,
Mm-hmm.
but that turned out to be better because I saw that the root length varied. But my parents actually got sick with the coronavirus, so I had to keep up shuttling them outside and inside and watering them during that time. So that was hard, but
Plus you were taking care of your sisters.
yes, plus I was taking care of my sisters. But I think overcoming them just kind of came naturally. I didn't have to do anything really, because I still got results, cooler results, I think, and they grew. They still grew. It wasn't like they never sprouted or anything.
Okay. What questions, if any, were created as a result of your work?
Well, I really wish there was a way to actually see the DNA sequence of the radish, but obviously that didn't happen. I think that would be really cool, and so I wondered about that, and I wondered if the seeds embryo, because the non-organic radishes sprouted sooner than the organic, but I wonder if the non-organic embryo was kind of not worn out, but a little bit, like chemicals getting at it, and therefore it sprouted quickly. But even though it sprouted like a day earlier, the organic seeds were still longer.
Interesting. Okay. Is there anything else that you want to add?
No, that's it. Thank you so much for watching.
Good morning. It's my pleasure to introduce myself and present my project to you all. I thank you for your time. My name is Divyam Signal Mathu, and I'm in 7th grade at Santa Rosa Academy. My science fair project is based on the question, how does the concentration of glucose solution in water on a plant affect its development? And I tested this with 4 different concentrations of glucose to water. This included the control group, also called 0 to 1, 1 to 5, 1 to 3, and 1 to 2. I originally got this project idea from a conversation I had with other students participating in the science fair and Mrs. Jessica Wood. We were discussing ideas in a meeting, and someone talked about growing plants in a dark place. This intrigued me as I knew that plants needed sunlight for a type of energy or sugar called glucose. I also wondered if they could be grown without sunlight, but only supplemented glucose. After more research in this topic, my question slowly morphed into, how does the injection of glucose affect plant growth? After speaking with the SRA horticulture teacher, Mr. Robert Montoya, I understood that the vascular tissue of many plants is too thin to accurately inject into, so I adjusted my focus to watering plants with glucose solution instead. I was very curious to see how supplementing this energy might affect the growth of plants in general, but what interested me about this overall subject is whether or not it's possible, since they're the only living things that produce their own food, to help expedite their growth by providing another source of this energy. What also interested me further is the idea that providing extra sugar might positively impact their development, along with all other living things as a result. But my project isn't simply interesting to think about, it's also very valuable in today's ever-growing society. Many people who live in poverty do not have the funds for nutritional food, but experimenting with ways of naturally growing plants more efficiently may reduce this problem. If it is easier to grow larger quantities of food and other products, then it will be much cheaper, meaning more families will be able to afford better things and live better lives. Also, many parts of the economy that rely heavily on plant growth, such as cotton, timber, or paper, will have more growth and be more productive. Besides, if plants can grow faster, many problems like climate change will lessen as more carbon from the atmosphere will be stored in plants, leaving more oxygen, not carbon dioxide, in the air. However, the goal of this project, briefly said, is to understand if supplying plants with an external source of food might be a viable option of easily improving their growth and development. But this is easier said than done, and so I also encountered a few minor problems while conducting my experiment, so I thought I would share 2 of them with you. Because of the sugar in the solutions, the top few centimeters of topsoil in some pots became extremely tough and sticky. This is a problem for any sprouts attempting to reach the surface, so I solved it by simply removing it from the pots that had it. Another slightly larger problem that I faced was the fermentation of the glucose solutions. To save time whenever I watered the pots, I created 1-gallon bottles with the different concentrations of glucose to water, but over time this caused them to ferment, and the compounds created by this process could kill plants. The fix to this problem was to mix and measure the solutions more often, and I did this about once every 5 to 6 days. I noticed this problem far into my project, so I'm not sure whether it may or may not have influenced the results, though I'll talk about this later on. Overall, my final measurements show that adding glucose to the water used in plants does not positively affect their development, but on the contrary, it hurts it. The control group grew normally, as you can see here, to an average length of about 14 centimeters, but the higher the concentration went, the less growth there was, as in this 1 to 2 pot. The 1 to 2 group overall had an average length of about 1.2 centimeters, and the higher concentrations, which included 1 to 3 and 1 to 2, never made it over the surface, but the slightly lower one, 1 to 5, did, and then died off. I searched through the soil for answers to this, and found that the 1 to 3 and 1 to 2 pots had many colonies of mold, and a hard top layer that suppressed growth, although there was some germination beneath the surface. I asked Mr. Montoya, the expert, for his opinion on why the 1 to 5 seedlings may have died, and he suggested that it was a fungal infection that killed them. If you take a look at my digital notebook, and I hope you do, you'll find data tables with precise measurements of the results, and graphs with a definite pattern in which more glucose caused less growth. In general, my project concludes that the more concentration there is in a plant's water, the less development there may be. When the control sprouted first, nothing else did for another week, I started to see a distinct pattern which caused me to have questions. One question I had was regarding the method of supplying glucose. Many articles I read proved that injecting different forms of energy into plants caused an increase in growth, but I was seeing the opposite trend in my glucose-watered plants. I wondered if watering the seeds and then once they reached the surface, injecting them would help their growth, but I never tested this, as it would require exacting precision to insert a needle into the vascular tissue of a plant. The major question that plagued me during my experiments was whether a lower concentration would help the growth of plants. I knew that the concentrations I tried were way too high, and I was very interested, so I tried a concentration of 1 to 10. My experiment results showed that it was better than other solutions I tried, such as 1 to 5 or 1 to 2, but still not better than the control group. And so if I were to do anything differently, which I most definitely would, I would measure and mix the solutions of glucose to water more often to prevent accidental fermentation. I believe this process of fermenting may have affected the results of my experiment by causing increased fungal and microbial activity in the soil, and so I try to stop from happening ever again. Another thing I'd do differently is start watering the glucose onto the plants once they're above the surface, so that they have a chance to live. This would allow me to measure exactly how much they affected growth, either slowing, stopping, or killing them entirely. Other than these 2 execution problems, I think I completed a very comprehensive and well done report. A few details that make my project distinctive is that every day of my experiment, I wrote detailed notes of what I observed into my digital notebook and spent many hours of hard work and effort in an attempt to make it the best it could possibly be. I spent a large portion of my time on this notebook, and it also requires a great deal of dedication and determination to wake up to water pots of plain soil and spend as much time as I personally have in an attempt to understand why things worked the way they did. This wraps up the video presentation of my science fair project. I thank you all for your valuable time and hope you have a great rest of your day.
Hello, my name is Savannah Smith. I'm in 7th grade, and my science fair project was to clean up oil spills in the ocean and on animals. I used motor oil and water to replicate oil spills in my kitchen, and all oil was disposed of responsibly. All animal samples came from the local sporting goods store and were not harvested for the purpose of this experiment. Overall, I did a total of 6 experiments, and I used different types of natural solutions to clean up oil off of animals and in the ocean. My first experiment was Dawn dish liquid soap on a sponge to clean up the water and oil mixture versus a dry sponge with no soap. The results was that Dawn was way better than the dry sponge. The Dawn ended up eating the oil more than it actually picked up. And that is very good because that way you don't have to dispose of a large amount of sponge. The Dawn ended up creating double the amount of clean water than there was before. I would definitely recommend this for cleaning up oil spills. My second experiment was Dawn on rabbit fur and deer hair. I soaked the fur and hair in oil for 30 seconds and 1 minute. Then I washed with the Dawn and let dry for 24 hours. The results were that Dawn cleaned the fur back to a natural state. This is good because we know that Dawn does work to clean oil off of animals, which is necessary in an actual oil spill. I would definitely recommend this for cleaning oil off of animals. However, when I did my third experiment, Dawn on duck feathers and chukar wings, I soaked the feathers in the wings for 30 seconds and 1 minute. Then I cleaned with Dawn and let dry for 24 hours. The results were that even though Dawn cleaned the duck feathers, they were broken and damaged, and the chukar wing was not clean between the feathers. Only the top saw any improvement. Unfortunately, this means that Dawn is not very effective for cleaning off birds, which can be dangerous because many birds get caught in an oil spill, and although we could probably clean off ducks if we were very careful, that would take a lot longer, and oil spills need to be contained in a timely manner. For my fourth experiment, I used peat moss as a test subject for soaking up oil. I tried it in both a water and oil mixture and with straight oil. My research showed that peat moss had been used before, so I wanted to try and recreate the results. My results were that 0 oil or water was measurably absorbed. I weighed the peat moss before and after a 30-second and 1 minute soak, and there was no difference in the weights. This experiment was a failure, so I would not recommend using peat moss to clean up oil. Next, I tested a spa sponge to see if it would be as effective on crude oil as it is on human oils that it observes in a spa. I cut the sponge into 1x2 pieces and soaked them for 30 seconds and 1 minute. I had to change my experiment after initial testing, as I found that the sponge would not absorb unless it is initially saturated. I changed my experiment so that I'd first push the sponge under the oil and water mixture, not holding it under or squeezing it, just submerging it for a second. My results were that changing my testing method made a huge difference. The sponge would absorb significantly more after being submerged. However, it has a low saturation point. It would not be overly effective with large quantities of oil. Even though I was able to increase my results dramatically, it was still a very small, about 5% compared to the 1% without being submerged, of the oil that was used. This would be more suited for small spills around the house. For my last experiment, I used human hair mats as a test for cleaning up oil. This experiment was based upon the Costco Busan oil spill, where scientists used doormat-sized samples of human hair mats to clean up the spill. They had incredible results, and I wanted to see if I could replicate it. I used hair mats that were 1 by 2 inches in size and soaked them in oil for 30 seconds and 1 minute increments. My results were that the hair soaked up 29% of the oil at 30 seconds and 45% of the oil at 1 minute. I was able to recreate these results with multiple tests, proving their accuracy. This experiment was as successful as the Dawn experiment. Overall, my first and last experiments were the most successful. They were both cost-effective and efficient. I believe that they are both great options to clean up oil spills in the ocean, if used together, and that together they will be the best solution. If I were to continue my experiment, I would try soaking the hair mats in Dawn, then using that to clean up the oil spills. I think that the Dawn would eat the oil, while the hair would soak up what remains at the same time. This could be the most natural, best solution that we have currently available. I hope you enjoyed my experiment, and thank you for letting me share my ideas.
Hello, my name is Braden Godfrey, and I'm in 7th grade White Track, and this is my science fair project. Every year, millions of people around the world get sick and die from dirty and unfiltered water. However, I believe that I may have found a solution for this problem. Let's look at my idea. Here I am on a website named Tinkercad. This website allows you to design anything you want, and then it allows you to convert it into a file to 3D print it. I only designed it and didn't make the actual unit because I don't have access to the correct resources. All you need to make this is a 5-gallon water jug, right here, a lightweight backpacking water filter, right here, a 1-liter water bottle, right here, a 2-gallon work bucket, right here, 2 small UV lights, right here, and then there's another one right here, 3 PVC pipes that are about 4 feet long, right here, and a 5-gallon camping jug with the spigot on it, right here. The device is very simple to make. All you have to do is cut the end of the 5-gallon jug and then attach the filter to the mouth of the jug. Then you attach the other side of the filter to the 1-liter bottle. After you do that, you have to cut a small hole in the lowest point of the 1-liter bottle so that the water slowly trickles out. This is where you put the UV lights, on opposite sides of the bottle as low as you can, as seen right here. Now you must slightly elevate the 1-liter bottle after cutting a large hole in the bottom of the 2-gallon work bucket. Once you do this, you have to cut one more hole in the top of the 5-gallon camping jug, the same size as the one in the work bucket. Then align the 2 holes. The last step is to secure the PVC pipe on the 3 sides of the camping jug that don't have a spigot. You are now finished building it. Here is the end product. The way that it works is that you pour the dirty water into the top of the upside down 5-gallon jug, right here. The water then goes down through the backpacking filter to clean out any small particles. Then it goes down to the 1-liter bottle where the UV lights disrupt any virus's DNA so that it can't reproduce, which is what makes you sick. Finally, it travels down into the camping jug where you can get the clean water from the spigot. Here, the PVC pipes are just to elevate the camping jug off of the ground so that you don't have to bend over to get the water. And this bucket right here, this work bucket, is to make sure that no debris goes into the filtered water after it's finished filtering. The whole project would cost about $250 to make, depending on which products you use. This is not including the UV lights because you would have to get the lights custom made. I looked everywhere, even on the website that has every single UV light in the world, and it does not have it. So, all in all, I think this is a great way to solve that problem of people dying from unfiltered water. Say this because it is very cheap and easy to make. You could make it yourself at home. Thank you for listening to my project. I hope.
Hello, my name is Isabel Marie Lumumba. I'm 6th grade Blue Track and I go to Santa Rosa Academy. For my project, I decided to do an engineering project. The goal of my project was to help the Philippines gather plastic and throw it away in the right place. The Philippines has a big problem with plastic. It is the third most biggest polluter in the world. I got my engineering idea from my mother. She told me that Philippines has a big plastic problem today, and I wanted to help the Philippines find a solution that would pick up plastic and put it in the right place. The thing that interested me most about this topic was when I was researching. I was looking at the other solutions for lessening plastic in the Philippines. One of the solutions I found was to make shoes out of plastic. My results at the end turned out to be good. My vehicle was able to scoop plastic that I put out on the floor. My invention is just like a garbage truck, but smaller. I used a vehicle that is popular in the Philippines, and it is called a tricycad. I wanted it to be a smaller version because there are narrow neighborhoods in the Philippines, and you can often see trash there. For the materials I used a block set that had wheels and sticks that could connect to each other. I also used cardboard so that the whole vehicle could stick together. So here's how my invention works. Once the vehicle enters the neighborhoods, there should be piles of plastic on the floor. So the vehicle would drive over the plastic, the scooper in the back would scoop it up and then dump it into the caddy. I was able to meet all my requirements. It can scoop the plastic and it doesn't fall out of the caddy. It is also really quick and easy to use, and it is way smaller than a normal garbage truck. An error that I have with my project is when the plastic dumps into the caddy. When the plastic turns around, it actually doesn't dump in the caddy. It dumps inside on the floor, actually. If I put the scooper closer to the caddy, it will hit the caddy and the whole vehicle will stop moving. If I were to fix again, I would definitely add a conveyor belt. When the scooper comes around, it will dump the plastic on the conveyor belt. The conveyor belt will then turn and then it will drop the plastic into the caddy. Another solution I came up was that if the vehicle went at a certain speed, the plastic in the scooper would be able to fling out and fly into the caddy. Unfortunately, my invention / model could not go like very fast because it is very fragile. So if I were to make this in real life, I would definitely have to make it very stable so that this solution would work. I think that this invention is applicable to the real world because tricycads are a popular form of transportation, and if this in the Philippines, and if this invention is added to the tricycad, this can help pick up plastic anytime. This is also a nature-friendly invention because the vehicle can be man-powered. Question I definitely have after this experience is, how can I improve the way the plastic goes into the caddy? I think this question would be very helpful if I ever create this again and want to improve it. Another question I have is, if this invention was made in real life, how will this invention affect how much plastic there is in the Philippines? That's all for my video presentation. I hope that you enjoyed learning about what I did in my project.
Hello Rangers, this is Mr. Cap, and I'm here to excitedly announce the winners of our 2020-2021 Science Fair. And just so you know, we conducted a small science fair, and we have a number of winners that I'm going to announce in just a minute. And our finalists will be moving on to represent us, Santa Rosa here, at the county level. And so we are so excited for them. So without further ado, it is my privilege to say congratulations. First of all, to our runners-up. And so that is Miss Coral Noonan, as well as Isabel Marie Lumumba. And so congratulations, runners-up. You guys did an awesome job. We are so proud of you. And that takes me to the drum roll. All right, our second place winner with an amazing science fair project. And a big shout out and congratulations to Jake and Benjamin Kirby. Well done, gentlemen. Well done. So, with that being said, final drum roll. The winner of our 2020 Science Fair, 2021, Science Fair is Divi Nalamathu. So congratulations to Divi. You did an amazing job. Congratulations to Jake and Ben Kirby. You guys were amazing. Congratulations to our runners up, Coral and Isabel. Incredible job. And all the rest of our participants, we salute you. We appreciate you. Great job on your science fair project. Awesome job, Rangers. Way to go and represent us well. Take care.